So thank you very much. So I'm going to tell you about some of our recent work where we've been interested in how neurovascular coupling might change in different brain regions. As the title says, and what that might mean for disease processes. So as I'm sure you know, neurovascular coupling is the process by which increases in local neuronal activity can cause local blood vessels to dilate and increase blood supply to different brain regions. And presumably this is important for the brain to match energy supply and demand, so the hungry neurons can get extra oxygen and glucose that they need when they're being really active. But as you also are, I'm sure, very aware, this is the physiological basis for the bold signal. So it's the big increase in blood flow that you get, an increase in blood oxygenation that you therefore get when blood vessels dilate, that gives you the positive bold signal that can be used as a surrogate of neuronal activity. And so obviously the relationship between what neurons are doing and what blood vessels are doing is really important for making sure that bold signals are interpreted kind of as appropriately as possible. And we know now quite a lot about the relationship between blood flow, bold signals, and neuronal activity in the neocortex. So we know what neurons are largely responsible for producing what signals to blood vessels, which blood vessels dilate, and how this blood vessel dilation can propagate up from the capillary bed into the PL arteries, giving you progressively bigger increases in blood flow. So we know quite a lot about that in neocortex. But importantly, we don't know very much about other brain regions. So for example, the hippocampus, and particularly subcortical regions as well, and the hippocampus. And that's because they're less accessible largely. So we know quite a lot about the cortex because we can look into it easily. It's on the surface of the brain. If you put a little hole into the skull, or if you use surface recordings, then you're recording from the neocortex, but you're not really sampling from deeper down. And so, but there's a number of reasons to think why neurovascular coupling might be different, and the relationship between neuronal activity and bold signals might be different in these deeper brain regions. And the hippocampus is obviously particularly important because it's known to be susceptible to hypoxic damage. And so understanding the relationship between its energy supply and demand would seem to be particularly important for understanding its function and pathophysiological processes that might be going on. But also because from a functional imaging perspective, Arnie Ekstrom's lab, for example, have shown that there's not a great relationship, not a very strong relationship between local field potential recordings and bold. So there's reasons to think the hippocampus might be different from neocortex. And so we wanted to investigate that further. And so the data I'm going to be showing you today comes from our Nature Communications paper that came out in June, so you can go and look at anything else and check it out there. And it was largely done by Kira Shaw, postdoc in my lab, who did most of the experiments. So what did we do? So we were interested in looking at neurons and blood vessels to understand the relationship between them. And to do this, we use mice that express fluorescent calcium indicators in neurons, so we can see neuronal calcium changes. I'm going to just play these two little videos so we can see how neuronal activity is changing, and then we inject a fluorescent dye into blood vessels and we can see blood vessels constricting and dilating. So hopefully you can see that here. This is a big vessel. It's kind of showing vasomotion. It's getting larger and smaller, and you can see the neurons are flashing. And so we put a cranial window into the skull, and in the case of the and access the visual cortex or the hippocampus. And in the case of the visual cortex, that just means removing a little circle of skull and putting a little kind of glass window in. In the case of the hippocampus, we have to aspirate the overlying cortex and put in a cannula through which we image. And so that allows us to see the hippocampus or the visual cortex. And then we can show the mouse. So the mouse is awake. It's on this cylinder. It can run if it likes, or it can stay still. It's up to the mouse. And then under a 2-photon microscope. And we can show the mouse visual stimuli in front of it on computer screens. So it can navigate through a virtual reality maze, or we can just show it basic visual stimuli, so drifting gratings. In addition to 2-photon imaging, which allows us to measure individual cells activity and individual vessels constriction and dilation, we also don't seem to be moving on. There we go. We also have a hemoglobin, combined hemoglobin spectrometer and laser Doppler flowmeter, which allow us to measure kind of more regional hemodynamic responses. And so this is shown here for recordings from the visual cortex. It's nice and easy. We can show them a visual stimulation and then stuff happens, which is less easy to do in the hippocampus. And so what you can see here is that we can measure the oxygenation state of hemoglobin. So we can see oxyhemoglobin here, deoxyhemoglobin here. We can also measure cerebral blood flow with the laser Doppler flowmeter. And we can, from the oxygenation state of hemoglobin, we can get the oxygen saturation of the blood, the SO2. From these values, we can also calculate the brain oxygen consumption, CMRR2. So we can get a number of different measures about what's going on from a kind of neurovascular perspective, using the 2-photon imaging experiments or using these VMS oxyprobe experiments. This is the name of the thing. It comes from Moore Instruments. And so what do we find when we compare hippocampus and visual cortex? Well, for a start, looking just at the kind of gross hemodynamic measurements, we find that the oxygen consumption rate is equivalent in the two brain regions. But in fact, the blood flow, the cerebral blood flow that we see in the hippocampus is lower than in visual cortex. And the blood oxygen saturation is also lower in the hippocampus than the visual cortex. So why could this be? Well, one reason has been known before, but we also showed it here as well, is that the capillary density in the hippocampus is lower than in the visual cortex. And so this is now an image from a post-mortem image from a mouse that we've perfused with a fluorescent gelatin. And you can see the pericytes, these little contractile cells on capillaries, which are in red, just because it's a transgenic animal that expresses a red fluorescent protein in pericytes. So you can see in the hippocampus and the visual cortex, and you see that the visual cortex is denser, and that's statistically significant. But that's not the only thing that's going on. In addition to this lower capillary density, we also see, if we look at individual vessels and blood flow, or flow of red blood cells in individual vessels, using the 2-photon microscope, we can see that there's other changes as well. And so what we do to do these experiments is we scan very fast along and across individual capillaries. So this is a capillary that's filled with a fluorescent dye, and the black streaks are red blood cells because they don't take up the dye is in the plasma. And so what we can see is, as we scan very fast along and across the vessel, we get something like this, this image here, where space is along the scan path is in this direction and time is in this direction. And that allows us to measure the diameter of the vessel, but also the red blood cell velocity. And that's because these black holes that are red blood cells, if they're going really fast, then they would move quickly along here. So they would have gone a long way in this dimension in a short space of time, whereas if they're going more slowly, then they progress more slowly across here. And so you get a sort of steeper stripe on the line scan image. And so we can tell from the angle of these stripes how fast the red blood cells are going. We can also count how many there are, and we can see how much of the volume of the blood is taken up by red blood cells as well. And so when we do that, we can see that the hematocrit, so the volume of red blood cells, is lower in the hippocampus than the visual cortex. The flux of red blood cells, so the number going past per second, is also lower in the hippocampus than the visual cortex, and the red blood cell velocity is also slower in the hippocampus than the visual cortex. So it's not just the case that the blood flow and SO2 in the hippocampus are lower because there's fewer vessels, but also because there's lower red blood cell flow in each individual vessel. And that results then, because blood cells are going through capillaries more slowly, that more oxygen can be extracted from each red blood cell to sustain this equivalent level of CMR2 in the visual cortex and the hippocampus. So so that sort of makes sense why we would get a lower SO2, because you've got this lower flow in each individual vessel. So that's kind of the baseline state of affairs, but we also wanted to look at how responsive vessels are in hippocampus versus in visual cortex. And to do this, we're now using 2-photon imaging of kind of XY movies. So we've got a vessel and we record this field of view over time. And you can see here's a vessel that's loaded with Texas red dextran. And there's baseline, there's not very much calcium signal in the surrounding neurons, but then the calcium signal goes up and down. And so what we can do is we can look at when the calcium signal in the local neurons goes up. And so you can see this trace here. So it spikes occasionally, and we can detect those peaks. And then we can see what the blood vessel was doing just after those peaks in local calcium. And so when we do that, we can see whether or not the vessel dilated after a peak in calcium. And so then we can define how many vessels were responsive or not. And what we find is that hippocampal vessels were less responsive to changes in local calcium than visual cortex ones. So you can see that quite a few fewer responsive vessels in the hippocampus. And then if we look at the sizes of those dilations when they do occur, you can see that even though the peaks in the calcium were very similar between hippocampus and visual cortex, when vessels dilated in the hippocampus, the dilations were smaller in the hippocampus than in the visual cortex. So the visual cortex, sorry, I should have said, is always orange and the hippocampus is always purple in these plots. So not only is blood flow and oxygenation lower in the hippocampus than visual cortex, but also the blood vessels in the hippocampus dilating less to local neuronal activity in the hippocampus than in the visual cortex. And that could be for a couple of different reasons. It could be because the neurons are less able to, and astrocytes are less able to send signals to blood vessels, or it could be that the blood vessels themselves are less able to dilate. And to cut a long story short, in the interest of time, we think that the reason that the hippocampus responses are different from visual cortex is because the vasculature is different rather than the neuronal signals are different. And that's for a number of reasons. One is that we didn't see any differences in neuronal activity patterns. So we looked at the kind of degree of correlation of neuronal activity and the size of those peaks and didn't find any differences. We also looked at existing single-cell mRNA databases of transcriptomes of different cell types and found that when we looked for neurovascular signaling molecules in neurons and astrocytes, there was no difference in, no signaling molecules were expressed less in the hippocampus than the visual cortex. So that couldn't explain the differences we see in terms of neurovascular coupling. What we did see using these single-cell mRNA-seq databases was that there was a lower expression of vascular functional markers in neural cells, so pericytes and smooth muscle cells on and endothelial cells. So vascular cells showed a lower expression of things that would be likely to mediate dilation. And so an example of that is this KCNJ2. Now this is a gene that codes for inward rectifying potassium channel. And that has been shown to be, by Mark Nelson's lab, has been shown to be really important for propagating a dilation from the capillary bed upstream to those big arterioles that would generate a large blood flow response in response to neuronal activity. And so it would sort of be consistent with the smaller responses we see if there was a problem in propagating dilation in the hippocampus compared to the visual cortex. Another thing that was different, and there are a few different ones, these are just ones I've highlighted, you can see the paper for all of them, but was a prostaglandin E synthase in endothelial cells, which was found to be pretty much absent in the hippocampus compared to visual cortex. And prostaglandin E synthase produces prostaglandins, which dilate blood vessels. So this is again a target that would be expected to be promoting dilation, but seems to be absent in the hippocampus. We also looked at the morphology of the pericytes that are responsible for constricting and dilating capillaries in the two brain regions. And we found morphological differences between the two. So we found that the pericytes were longer in the hippocampus than the visual cortex. So the distance between pericytes was greater, and the cell length was longer, particularly of the pericytes. These are mesh and thin strand pericytes that are found in the middle of the capillary bed, and they were longer. So there were some morphological differences in pericytes, and there were these kind of expression differences between the two brain regions. And this pattern suggests a different vasculature or different vascular physiology in the hippocampus than in the visual cortex that's consistent with it being less able to dilate. So pericytes that are the most contractile pericytes are shorter and have lots of processes that wrap around the vessel. And so it's possible, and pericytes tend to constrict more and dilate more near the soma. So it's likely that having longer pericytes means that the vessels are less able to constrict and dilate, or at least it's consistent with that. So we would argue that we would argue that these vascular differences are what underlie the neurovascular coupling differences that we see between the weaker neurovascular coupling that we see in hippocampus. But we wanted to understand what these differences that we've observed would mean in terms of oxygen delivery to cells in the hippocampus versus the visual cortex, and what that would mean for for their ongoing function, and whether this might explain the hippocampus's susceptibility to hypoxia. And so what we did is we took a in vivo, we basically mapped the vascular network in vivo. So this is an image of a Z-stack of the vascular network in one of the brain regions, I forget which, sorry. And then we took that data and we converted it into a distance map where we found how far any sort of brain, any parenchymal pixels were from a blood vessel. So we could basically get a distribution showing for the two brain regions what sort of how many pixels were close to blood vessels and how many were really far away. And so then we could look at the tissue that's on the average distance, the median distance from a blood vessel, versus the kind of the tissue that just happens to be, because of the irregular vascular network in brain, that some tissue will happen to be relatively far from a blood vessel compared to others. And so what we did is we took the readings that we've got of blood oxygen saturation in the two regions and the vascular densities that we've recorded, and we modelled oxygen diffusion into the tissue from using these values. So here's a model capillary, and we model oxygen diffusion out into the brain from a capillary. And when we do that, we can measure the predicted oxygen, we can predict the oxygen gradient that occurs in the brain tissue as you go away from the vessel itself. And again, orange is visual cortex and purple is the hippocampus. And we've done this for two different predicted rates of oxygen consumption by the brain. So because the brain's consuming oxygen, as oxygen diffuses into the brain, the concentration drops. And so because the hippocampus has a lower starting oxygen concentration than the vessel, it starts lower down. But actually, if you look at the average distance from a capillary at the median, there's not a massive drop even in the hippocampus. So there's a gradient, but it doesn't drop to too low levels. And if we predict what that would mean for oxygen consumption and ATP production from oxygen, so the rate of respiration, we can see that actually we predict that the median distance from a blood vessel, that the oxygen consumption rate, so ATP production, remains above 90% of what it would be at max. So in this condition, oxygen concentration isn't limiting for ATP production. But that's not the case if we look at the small proportion, the 5% of the tissue that's the furthest from a blood vessel. So just because of the irregularity of the vascular network, some of the tissue will be further from a blood vessel. And so we look at that 95th centile, and we look at the highest, which is actually likely to be the most physiological rate of oxygen consumption. We can see that in visual cortex and in hippocampus, there's now quite a sizable gradient of oxygen concentration away from the vessel. But even in the visual cortex, this doesn't really impact on respiration rates, because oxygen doesn't drop low enough to start become limiting for ATP production. But in the hippocampus, that's not the case. And oxygen now, in that furthest part of tissue from the vessel, does drop to low enough oxygen concentrations where it starts to inhibit ATP production and oxygen consumption due to oxidative phosphorylation. So in this part of the tissue, we suggest that there's about a 40% decrease in ATP production, potentially due to the oxygen concentration. Now, this is only a tiny part of the tissue, and obviously the hippocampus can normally deal with this. But what it does suggest is that the hippocampus is much more, compared to visual cortex, at a watershed where ATP production is starting to be inhibited and oxygen is quite low. So that suggests that it wouldn't be that difficult to push it into a pathophysiological situation, where if you started to reduce oxygen levels in the blood even more, that then you might hit pathology. And that would be consistent with the known susceptibility of the hippocampus to hypoxic damage. So just to summarise this part of the talk, I've shown you that there's lower blood flow and lower blood oxygenation in the hippocampus than in the visual cortex, and that there's also weaker neurovascular coupling in the hippocampus than the visual cortex. We think that's because there's differences in the vasculature, because we see changes in vascular functional markers and changes in vascular cell morphology in the hippocampus compared to visual cortex. And that we predict that this means that oxygen is near a watershed where it's limiting for ATP production in the hippocampus, and it's much further away from that position in the visual cortex. And so this suggests not only that the hippocampal neurovascular coupling properties and the relationship between neuronal activity and the bold signal is likely to be different in the hippocampus than it is in visual cortex, but also that these differences in vascular function might put the hippocampus at risk of damage when oxygen levels in the blood or vascular function decreases. So this is a kind of potential pause point if anyone would like to ask any questions, clarification on the first part of the talk. I'm subsequently going to consider a bit more the potential role of these pathways in pathology. Yeah, so basically we're interested in whether this kind of difference between this regional difference or this susceptibility of the hippocampus might be important for disease states. And so one of the disease we've been focusing on has been Alzheimer's disease, and that's because it's been known for some time that risk factors for Alzheimer's disease are shared with cardiovascular disease, which suggests there's a kind of cardiovascular component to the disease. And more recently, it's been shown that blood flow changes, in fact, precede in humans, that blood flow changes precede more classic Alzheimer's pathology. So you get changes in blood, decrease cerebral blood flow before you get accumulation of beta amyloid, and that happens before structural change and cognitive decline. So obviously here the decreases in blood flow are particularly interesting because they're the first sign that something's going on and likely to be causative in some way. And so obviously what cerebral blood flow changes might be doing is decreasing the energy supply while not presumably in normal physiology affecting the energy use, or the oxygen use by the brain. So what might well be happening is that you're getting a decrease in brain oxygenation in these conditions where you've got a decrease in blood flow. And that's really interesting or potentially relevant because it's been shown in vitro that hypoxia can promote tau phosphorylation and beta amyloid, decrease beta amyloid breakdown and increase beta amyloid production. And so potentially hypoxia could produce the plaques and tangles that are classically found in Alzheimer's disease. And so this has led to the vascular hypothesis of Alzheimer's disease that's been proposed originally by De La Torre, but expanded on by Zlokovic, Ostergaard and others, whereby cardiovascular risk factors lead to decreases in brain blood flow, brain hypoxia, and then beta amyloid accumulation and neuronal toxicity. Because beta amyloid accumulation can itself reduce brain blood flow, you can get a vicious cycle whereby you get progressively more brain hypoxia, more beta amyloid accumulation, and obviously lots of other pathways kicking in as well as pathology kicks in. So what we've specifically done is we've asked whether the main genetic risk factor for Alzheimer's, ApoE4, one of the things it might be doing is to kind of engage this pathway to decrease blood flow and increase brain hypoxia. So what we've essentially been interested in looking at is whether ApoE4 might be affecting neurovascular function. And so some of the data I'm going to show you is available on bioRxiv as a preprint. So we've done this study kind of twice in visual cortex and in hippocampus, with slight necessary differences in methodology in the two regions, as I'll explain. But essentially, and the visual cortex data is on the bioRxiv paper, but we're hoping to submit an updated version with everything very soon. So first of all, we looked at capillary density again to see if there were differences in ApoE4 versus ApoE3 transgenic mice. So these are mice that don't have the murine version of ApoE, and instead they have either human ApoE4, the genetic risk factor for Alzheimer's, or ApoE3, which carries a normal Alzheimer's risk. And what we find is that there is a decrease in vascular density due to associated with ApoE4, the ApoE4 gene. So there's a lower vascular density in both the hippocampus and the visual cortex. And it seems to be particularly decreased in the hippocampus. But we actually don't find very, very large differences in kind of baseline hemodynamics. So what we see is we see that there's no differences in red blood cell velocity, there's no differences in hematocrit. There's a slight difference in the variability of flow, which is associated with some, which has in other studies has been associated with some problems for oxygenation, so a decreased oxygen supply, but they're not very dramatic. We don't find any significant, sorry, I haven't run the stats on this, but I'm showing you the stats on this, but it's not significant decrease in red blood cell, in cerebral blood flow or in SO2 though. So these changes are not dramatic, they're not causing big effects that are big enough to cause a difference in cerebral blood flow or in brain oxygenation at this stage, which is kind of not what we were expecting. And we also don't find any differences really in neuronal activity at rest either. There's no genotype differences in the number of calcium peaks, in the size of those calcium peaks, or in the kind of degree of correlation of firing. However, when we look in visual cortex, so in visual cortex, we can, as I mentioned before, we can present a very discrete sensory stimulus, and we can look at what happens to that stimulation, which we can't really do in hippocampus. And so what we can do there is we can look at the response to that visual stimulation. And when we do that, we see that the calcium actually in neurons goes up compared to those in more in the ApoE4 mice than in the ApoE3s. And that's consistent in the ApoE visual cortex experiments. We've been able to follow these mice for up to 12 months in some of the conditions, but for the calcium we've only got up to 6 to 7 months, but that seems to be consistent across those age points. And when we look at blood vessels and see how much blood vessels respond to this visual stimulation, we find that across the age span, ApoE4 arterioles are less likely to respond to that increase in visual stimulation, to the onset of visual stimulation, than our ApoE3 mice. So it's a small decrease, but it's consistent across the age time. That's not the case for capillaries, and whether red blood cell velocity goes up when you show the visual stimulation, that isn't affected by the genotype. But there is this subtle but consistent decrease in arterial responsivity. When we look at the size of those dilations, however, they're not different, they're not affected by genotypes. If the vessels do respond, then they respond the same amount than in the 3s and the 4s. But possibly the most dramatic finding difference was found when we look at vasomotion. So if you remember when I showed you the very first image, the movie, there was a big blood vessel that was kind of pulsing, and this is termed vasomotion. It happens often at 0.1 hertz. It's probably important for the sort of resting state functional imaging signals that people look at. So it's about 0.1 hertz, and what we find is that in the ApoE4s that's decreased compared to the ApoE3s. So this is the power at 0.1 hertz. You can see it's reduced in the 4s, which are in grey, versus the 3s that are in purple, and that's the same across the age range. Oh, sorry, I've hidden the signals. This is in the young animal. Oh, it does say. So in the young animals, the middle aged and then the older animals are all, this decrease is present in all of them, and it seems to be particularly severe in the oldest animals. So vasomotion has been thought to be important for supply of oxygen. I mean, and to reflect kind of the functional state of the vessel. So what it looks like we've got is something that's not, that the PL arterioles are working less well in these animals, but noticeably they're not reflecting this increase in neuronal activity that we saw. So we found that the neurons were more active and the vessels are, if anything, less active or not changed. So in this case, this genotype difference is reflected in a way a difference in neurovascular coupling, but it isn't sufficiently sensitive to detect the increase in neuronal activity that we see. But it's a relatively subtle difference, I should say that. So then we looked in the hippocampus. Now in the hippocampus, like I say, we can't just shine a one single stimulus on and look at the kind of net response to that, because you just don't get a synchronous effect. But what we can do again is the same thing I showed you before. So we can look at local changes in calcium and see what local blood vessels do. And if we do that, again, we find that the ApoE4 vessels are less responsive, as we saw in the visual cortex, than the ApoE3 vessels. And that actually now in the hippocampus is the case for small capillaries as well as larger arterioles. And so we find, again, this decrease in responsivity to neuronal activity. Again, though, however, if we look at the size of those vessels, they don't seem to be any different. And if anything, the ApoE4 ones are slightly bigger, but it's pretty noisy. As you can see, there's not very many that respond. So this is quite noisy. So essentially, if they do respond, if they can respond, they respond to the same degree, but they're less likely to respond. And again, in the hippocampus, we can't, because the vascular anatomy of the hippocampus is different, the larger feeding vessels are actually deeper in the tissue, and we can't reach them, we can't image them with the 2 photon, we can't get deep enough down most of the time. But we can look at a kind of indicator of vasomotion by looking at fluctuations in red blood cell velocity. So as the vessels are dilating, then that will change the speed of the red blood cells that are going through the vessels. And what we see is that there's a reduction again in the power of the red blood cell velocity fluctuations at the same vasomotion frequency. And in fact, there's also a reduction in the heart rate frequency, because obviously red blood cell velocity will also change with the heart rate as well. So that's this big difference here. But if we look at the vasomotion peak, then it's also much smaller in the ApoE4s than the 3s. So what does that mean? So basically what we find that there's increased calcium responses to visual stimulation in the ApoE4 visual cortical neurons, and we don't see any differences in neuronal activity in the hippocampus. But what we do consistently see is that the arterioles, PL arterioles in V1 and the arterioles and capillaries in hippocampus dilate less frequently in the ApoE4s versus the ApoE3s, and we see reduced vasomotion in both regions, as well as a decreased capillary density. So there's some relatively subtle changes that are quite consistent across the brain regions, but they're not in these young animals that only, well, not all of them are young animals, in these animals that don't express any other pathological changes, these are insufficient to cause changes in the blood oxygenation or in blood flow. And we don't see any changes over time. So it suggests that what ApoE4 genotype is doing is it's just it's got subtle non-progressive effects on the kind of physiology, but it is in a direction that would maybe exacerbate future pathology. And that's kind of interesting because, ApoE4 is a risk factor for Alzheimer's. It's not causative in itself. It presumably does require the interaction of other factors in order to push the system towards disease. And so we're looking at a few of those things. I don't want to go into them in very much detail because I'm running out of time. But the things we're looking at are particularly the effects of beta amyloid accumulation on these physiological changes we've observed, and also the effect of some environmental or lifestyle factors that might be expected to push the system towards pathology. So one of those is exercise. So we're very nice to our little mice. So when they've had surgery, we give them an exercise wheel and they can be active on that exercise wheel. We did realize that maybe this was actually helping the mice to be healthy mice that would show pathology. And so Sylvia, PhD student at my lab, has been running an experiment where she just compares the mice that have the wheel with mice that don't have the wheel, so don't have the potential for exercise and are also don't have that potential. And as you can see, we do find when we look at the responsivity of these PL arterioles and capillaries, now this is just in visual cortex now, that we do see that it seems like exercise does have an effect. So and there's a genotype by exercise interaction. So E4 mice that don't exercise have particularly badly responsive vessels, both in arterioles and in capillaries. And so that suggests that indeed it is the case that there's a sort of, the system is perturbed in a direction that it can be pushed to become exacerbated, that might then, you can imagine over time, push you to a system where you could end up with progressive and Alzheimer's pathology. I don't really have time to go into that, but we also see then, we start to see some changes emerging in the macroscopic measures of blood flow and oxygenation as well. And then the last thing I wanted to mention is that we're also now trying to see what does happen. We get to explicitly test what happens in a kind of more Alzheimer's-like model. So we've got mice that express ApoE3 or 4, and now we want to see what happens. We characterize their physiology, and then we've bred them with mice where we can control the expression of beta amyloid, of APP, the precursor for beta amyloid. So we can characterize the vessels in ApoE3s or 4s, and then we can switch on beta amyloid production and see if beta amyloid accumulates around the dysfunctional vessels and in hypoxic regions. So we can more directly test now this hypothesis that there might be vascular changes leading to hypoxia, leading to beta amyloid production. And so we've been very slowed down by COVID on this project, but we are able to, we've generated the mice and we can see that, that we can accumulate beta amyloid in these mice after we've characterized them at baseline. So we'll see what happens. So just to conclude, and particularly from a BOLD perspective, what I've talked to you about today, I've shown you that the relationship between neuronal activity and hemodynamic changes is different in the hippocampus and the visual cortex, and that implies that the BOLD signal in the hippocampus represents a different amount of neuronal activity than visual cortex. I've also shown you that genetic and lifestyle factors, so ApoE and exercise, can affect the responsiveness of vessels, and they may alter hemodynamics. And that would alter the reliability of BOLD as an indicator of neuronal activity in these different genotypes or in people with different exposure to different risk factors. And also what we're particularly interested in looking at in the future is whether these different factors are pushing the brain towards pathology, especially in brain regions such as the hippocampus that in normal animals are already near a watershed, we predict. So we're really interested in seeing if we can understand how pathology might start to develop in those animals, in those regions rather. So Rachel mentioned this before, just before I stop, I wanted to shamelessly promote our third Interpreting BOLD meeting, which will be, it's been postponed twice now, but it will happen. It's not in 2020 anymore. It's going to be in 2022. If you're interested in chatting to people and seeing some great talks about understanding the kind of physiological basis of the BOLD signal, or what, how experiments can be done to better understand or maximize the information available from the BOLD signal, please do either follow me on Twitter or go to the website here, Brain Energy Lab /Interpreting BOLD, and you can to register an interest, because it's going to be great. I think I've got a, we're going to be talking about cells and circuits underlying BOLD, variability in neurovascular coupling, and extracting functional connectivity information from BOLD. And it'd be really great to have some of the speakers, and we'd love to see you there. So do let me know if you're interested. So it just remains me to thank my lovely lab, particularly Kira, and I totally forgot to mention her by name, but Orla, who did the visual cortex ApoE stuff, and Sylvia, who's been working on the exercise stuff. So, and the funders, obviously. So thank you very much, and I'd be very happy to take any questions.